Thank you for standing by and welcome to the Arovella Therapeutics iNKT cell platform explanatory webinar and Q&A today. All participants are in a listen-only mode. There will be an initial presentation lasting for approximately 30 to 35 minutes, followed by Q&A based on questions received via email and those that come through live. If you do have a question you'd like to submit live, please feel free to do it using the Q&A function on the Zoom app, and we'll get to those at the end. Appearing on behalf of Arovella today, we have the CEO and Managing Director, Dr. Michael Baker, Senior VP of Development and Translational Medicine, Dr. Minnie Bharathan, and COO Nicole van der Weerden. To begin, I'll hand it over to Michael. Please go ahead.
Great, thanks Matthew, and thanks very much. I'm delighted to have Minnie and Nicole both on the call, but certainly thank you very much to everybody that's taken the time out of their schedules to dial in today, and we are very much looking forward to taking you through the Arovella story. So we will be making forward-looking statements, so please do take note of our disclaimer. And I'd like to start with the highlights of the company. So most of you may know already, but if you don't, we are working on what we call an invariant natural killer T cell or iNKT cell platform. And what we know from that is it's going to be allogeneic, which means off the shelf. So it's an important treatment modality for various cancers, and we're currently targeting two targets, CD19 and also a DKK1 peptide for both solid tumours and also hematological malignancies or blood cancers. Now, we have world-leading partners through our collaborations and partnerships with Imperial College London, MD Anderson Cancer Centre, and also an exciting strategic partnership with Imugene, combining our platforms. Now, we do have a very strong leadership team, and I'll touch more on this in a moment, certainly with a lot of expertise in drug development, but also particularly around cell therapies. Now, as a company, we're very data-driven, and I think that was some of the reasoning for this webinar. We'd like to talk in a little bit more detail about some of the data that we've generated, because we do think it is quite exciting, but we certainly use it to drive our decision-making, both for key assets that we'd like to bring into the company, but also the clinical indications that we will tackle. And so we have, and we will continue to look for strategic assets or acquisitions that we think align well with our focus areas and fit with our current pipeline. And so as it stands today, we do think that we are one of a handful of companies globally working on an iNKT cell therapy platform, and the only Australian company working with an iNKT cell platform. So we do think that we are quite unique and well positioned for growth. Now, I'd like to start giving a bit of a background on CAR-T. Many of you will have heard this term, either cell therapy or CAR-T, before. And the T comes from the fact they use what we call T cells. So they're our most common type of immune cell in our bodies, that their natural role is like soldiers. They'll go around and fight infections or even particular types of cancers. So what CAR-T actually does, we use the term here autologous, which means from the patient. We actually take immune cells or blood from a patient, collect the immune cells using what's called a leukapheresis step, and then we genetically reprogram those immune cells so they produce something that's called the chimeric antigen receptor, or CAR. And I'd like you to think of that like something that sticks out on the surface of the immune cell as a guided missile that actually directs or drives these immune cells to go towards the cancer and trigger its destruction. So they get grown up into the millions, and then eventually when they have been grown and shipped around the world and back to the patient, they will be infused. And once that's happened, they'll be able to find these particular markers on the surface of the cancer cell. And once they do that through those chimeric antigen receptors, we trigger cell killing of the tumors. So in short, it has revolutionized the way we think about cancer treatment, because we are starting to use the word cure and cancer in the same sentence, which is a wonderful place to be. And what we've seen over the course of 2022 was a series of media articles culminating around research reports where patients that had taken these therapies more than 10 years ago, both adult cases and pediatric cases, they were still cancer-free. And for the two main products that were approved in 2017, Yescarta and Kymriah, in 2022, one did just over 1.1 billion in revenue and the other one over 500 million. And Abecma, another more recently approved cell therapy, was approved only recently and did just under $400 million in sales. So already generating strong revenue, and certainly the market is expected to reach double-digit billions by 2030. So we know, though, that there are some limitations of the current cell therapies, and one of those, as I mentioned a moment ago, is the fact that they are autologous. And that's the concept that the cells must originate from the patient that needs to be treated. So what this means is every single dose of the product needs to be manufactured bespoke or for that single patient. And what that does is it drives up manufacturing challenges and logistics. And of course, that in turn drives up the costs of these, not just from a manufacturing point of view, but the quality control and quality assurance. Now, in addition, we believe that because the starting material comes from the patient that's seen cancer or various forms of treatment like chemotherapies and so forth, you may actually have a less efficacious starting material. Now, also, there's only a limited number of centers that can actually collect these cells to go on to manufacturing. So not all eligible patients will actually receive the therapy. Now, in addition, it takes in the order of about 4 to 6 weeks for a patient to actually have that therapy manufactured. And so in some cases, patients do actually die while waiting for that treatment. And of course, because it's made every single time, you're never away from the possibility of a manufacturing run failure for a particular patient. So we do believe that our allogeneic iNKT cell therapy platform has the potential to alleviate some of these manufacturing challenges and have a big impact on the sector, but also from the preclinical data that we've got, we're excited that we may have a more efficacious product. Now, I just want to talk about some of the advantages of iNKT cells briefly. So, as I said, we've got 3 major cell types in the immune cell complement that we've depicted here: T cells, NK cells. Now, T cells sit on what we call the adaptive arm of the immune system. NK cells are on the innate side, and iNKT cells, which is the focus of our platform, bridge the middle and have actually properties from both. So we know from T cells is they have previously been shown to cause severe side effects like cytokine release syndrome and neurotoxicity when armed with CARs. And to get away from this, you need complex gene editing tools and techniques to remove what's called their T cell receptor. And we've seen for NK cells that there is also, even though they can be dosed off the shelf, which is a good thing, we're starting to see that there is potentially limited persistence of the cells, and that may limit the durability of the response. So for iNKT cells, on the other hand, they don't cause graft versus host disease. So this is really important. And what it means is that we can actually give the cells from a healthy donor to a patient. They naturally target tumor cells through what we call their invariant T cell receptor, which is depicted here in red. And when we arm them with a chimeric antigen receptor or the heat-seeking missiles I mentioned a moment ago, they're dual targeting. And we know they have the direct killing tumor mechanisms from both T cells and NK cells. Now, in addition to that, iNKT cells also modify the tumor microenvironment and actually kill some of the cells that actually promote tumor growth. They also infiltrate tumors, and once they've activated, they release a range of signaling molecules which can actually tell other immune cells like T cells and NK cells to get involved in killing the particular tumor cells. Now, in addition to that, so we believe there are significant advantages to working with an iNKT cell, but there's also another really important aspect, which is how crowded the sectors are. And so for T cells, this is a representation of some of the companies that are playing in this space. You can see that there's quite a lot, and it's the same story when we look at NK cells. So they are quite crowded versus, as I mentioned a moment ago, for iNKT cell therapies, Arovella does sit as one of a handful of companies that are actually working on these types of cells for immunotherapy platform. So with that, I'd just like to hand it over to Dr. Minnie Bharathan. But before I do that, I'd just like to point out that Minnie's got a phenomenal background. She was formerly a practicing veterinarian before completing her PhD in immunology. She's worked for some pretty high profile cell therapy companies, and most of her career has focused on this sector. And certainly when Minnie was joining, I think one of the key excitement factors for her was she'd already worked on T cells and NK cells, but she did see the benefit of working on an iNKT cell therapy platform because of some of those advantages. So without further ado, Minnie, I'll hand it over to you to take us through some of the science.
Thanks, Michael. And hello, everyone, to the listeners of this webinar. As Michael mentioned before, Arovella's platform technology is based on the use of invariant natural killer T cells for allogenic off-the-shelf approach. In fact, iNKT cells are T lymphocytes with an invariant T cell receptor. They also co-express natural killer cell markers. And in fact, these two properties makes them more potent than the conventional T cells or NK cells. So let me give you a bit of background on why this particular cell type is of importance in patients with different types of solid tumors. And here we are showing two studies that assess the iNKT cell frequency in blood or tumor as a prognostic factor in head and neck cancer and primary colorectal carcinoma. As you can see from the slide, the graph on the left side shows the correlation of circulating NKT cells to the improved outcomes for the patients with head and neck cancers. This is a prospective study where circulating iNKT cell numbers were evaluated in 47 patients before radiotherapy. And then these patients were monitored for 5 years to look at the survival benefit. And as you can see from the graph, the higher the levels of iNKT cells in the blood, the better the disease-specific survival of these patients. Similarly, the graph on the right side shows the survival rate of colorectal cancer patients in correlations with the iNKT cell infiltration within the tumor and was monitored over 8 years. And this case also, the greater the infiltration of tumor with iNKT cells, the better disease-specific survival of the patients compared to the low iNKT cell infiltration. So higher frequency of iNKT cells in tumor can be an independent prognostic factor for a favorable prognosis for these patients. And what does these data sets points to? And it points to the fact that the iNKT cells are rare, but unique and important subset of immune cells that could impact negatively on the tumor growth. Next slide, please. So how do we make allogeneic off-the-shelf CAR-INKT cells? And what are the advantages of this approach? So unlike conventional T cells, iNKT cells that we use in our platform are not alloreactive, as Michael mentioned before, and therefore can be generated from allogenic donors for rapid infusion into numerous patients without the risk of graft versus host disease. And iNKT cells could be used for allogenic use without any modification or minimal modification, unlike conventional alpha-beta T cells. And iNKT cells are known to naturally suppress graft versus host disease. So it is a perfect type of cells to use for allogenic approach from a graft versus host disease perspective. So coming to the manufacturing, briefly, iNKT cells are isolated from peripheral blood of a healthy donor. And to note that frequency of these cells in the peripheral blood accounts to about 0.01 to 1% of the mononuclear cells, unlike the T cells or the NK cells. Once isolated, these cells are engineered to express a CAR on the surface of the cell. And these CAR-empowered cells are then expanded, and at the end of the process, the cells are frozen down and stored. And the final product created from a single donor can be used to treat multiple patients. And when a treatment decision is made for the patient undergoes lymphodepletion, followed by infusion of this off-the-shelf product stored at the clinical site without any delay, unlike that happens in autologous CAR T cell therapies. And in fact, this methodology comes with several advantages. One, since we are starting with healthy cells, we expect to have more potency for the cells to perform in the recipient and hence better efficacy in patients. The second point is that it is scalable, meaning that we can treat several patients from one single manufacturing run, which potentially reduces the cost per patient. And since the third point is that it's readily available at the clinical site, so no waiting period is necessary for the treatment. And as there is no, since it is an off-the-shelf product, so no risk that there is a manufacturing failure and a patient will be in a situation where there is no product available for treatment. Next slide, please. So this is the schematic representation of how we manufacture our lead candidate, CAR-19 iNKT cells, or ALA-101. From now on, I will call CAR-19 iNKT cells as ALA-101. So in order to maximize the potential of ALA-101, iNKT cells with a particular phenotype are isolated from the blood of a healthy donor. As I mentioned earlier, the frequency of the cells are comparatively low. We modify a very small number of cells using small quantities of expensive reagents, like for example, the viral vector that we use to transfuse the cells. So once the cells are isolated, The cells are then engineered using an antiviral vector to express the CD19 CAR. And the engineered cells are expanded in the culture, and the cells are frozen down and stored until the experiment. So the graph on the slide shows the expansion potential of these cells on a research scale. And y-axis of the graph is the fold increase of cell number on a particular day, and x-axis depicts the days in culture. And as you can see, a 5,000-fold increase in the number of cells by day 21 of the culture. And we believe that in a larger scale manufacturing batch, we may be able to increase the fold of expansion to many more than what you're seeing in the graph. Next slide, please. So what are the mechanisms of action by which these iNKT cells kill the tumor? So once we engineered the iNKT cells with a chimeric antigen receptor, unlike allogeneic CAR-T cells or CAR-NK cells, CAR-INKT cells can destroy the tumor through multiple mechanisms. And these cells possess multiple receptors that could kill the cancer cells, one through the engineered CAR, and through the CAR, these cells recognizes the tumor antigen that is targeted by the CAR. And second, through the ITCR or the invariant T cell receptor, and these cells express TCRs will recognize the glycolipids that is presented on non-polymorphic CD1D, or indirectly by the CD1D on activated antigen-presenting cells. 3, by the direct killing of the tumor cells through the innate receptors. So in fact, it is a triple targeting mechanism compared to the other 2 cell types that we mentioned before. So in this process, activated NK T cells produce several cytokines with the capacity to jumpstart and modulate an adaptive immune response. Some of these activities include either blocking or killing of myeloid-derived suppressor cells or tumor-associated macrophages. And these cells also recruit and activate other immune cells such as dendritic cells, T cells, and NK cells in order to restructure the tumor microenvironment to an immunostimulatory one. Next slide, please. So iNKT cell platform can be engineered with a variety of CARs. Here is an example of iNKT cells engineered with a CD19 CAR, killing CD19 positive tumor cells in vitro on the left side. So in this particular testing, tumor cells and CAR-19 iNKT cells are cultured together in a tissue culture dish and then analyzed for the remaining live cells in the culture after 20 to 24 hours. What you can see on the y-axis is the percentage of dead tumor cells, which is expressed as cytotoxicity percentage, and x-axis shows the different ratios of CAR-19 iNKT cells or CAR-T cells to CD19 positive tumor cells. So one thing to clearly note in this graph is that the CAR-19 iNKT cells from 2 donors, top red line, are killing CD19 positive tumor cells better than the CAR-T cells. Similarly, on the right panel, the CAR-INKT cells are engineered with another CAR targeting another protein on the tumor cells. So you can see the CAR-INKT cells killing tumor cells that express the cognate antigen in this case as well. Next slide, please. So our lead candidate that is targeted to enter the clinic is ALA-101. The iNKT cells are genetically modified to express a CAR targeting CD19 on the tumor cells. And CD19 is an antigen that is expressed in normal B cells and malignant B cell blasts of leukemias and lymphomas. And CAR-T cell therapy is a proven therapeutic approach for these kind of leukemias and lymphomas. Similarly, CD1D is expressed in several tumor cells, including lymphoma and myeloma. As I mentioned before, iNKT cells can target the cells through the ITCR CD1D axis. However, CAR-T cells and NK cells are not capable of targeting through CD1D. And graph on the slide is depicting the effect of CAR-19 iNKT cells killing K562, a chronic myeloid leukemia cell line that lacks both CD19 or CD1D, which is in the blue line, and K562 cell that express CD1D alone in green, or CD1D alone in orange, or expressing both CD1D and CD19, the red line. So y-axis is the cytotoxic activity, expressed as percentage of dead cells, and x-axis shows different ratios of ALA-101 to tumor cell line used in this assay. As you can see from the graph, when the tumor cells express both CD19 and CD1D, the killing efficiency of ALA-1 increased compared to that of controls, suggesting that the additive effect of the anti-tumor activity through the CAR and the ITCR. Next slide, please. So our next test was to see the ability of the ALA-101 to kill patient-derived primary tumor cells. For this test, what we did was we collected primary tumor cells from 2 mantle cell lymphoma and 2 marginal zone lymphoma patients. As I mentioned for the previous graph, Y is the cytotoxic activity and x-axis shows different ratios of ALA-101 to tumor cells. And top panel shows the efficient killing of mantle cell lymphoma cells by ALA-101 over CAR T cells, and the bottom panel shows the killing of marginal zone lymphoma by ALA-101 at all effector to target ratios tested. We also tested killing of chronic lymphocytic leukemia cells from 2 patients by ALA-101, and we observed the similar results, and the data is not showing because of the time constraints. Next slide, please. So now that we have shown that ALA-101 was able to kill tumor cell lines and primary cells from lymphoma patients, our next question was whether it has been translated to in vivo in a mouse tumor model. And our hypothesis was that ALA-101 will kill the tumors faster and more efficient than CAR T cells. To test this hypothesis, we used NSG mice, a strain of mouse that would permit the growth of human cells. And graphic on the top of the slide is showing the experimental design. Here we used the cell line C1R-CD1D, which is a tumor cell line that expresses both CD19 and CD1D. In addition, these tumor cells also express a luciferase gene that would enable to visualize the tumors in mice as bioluminescence. And the tumor cells were injected intravenously to the mice 10 days before the treatment with cells. And tumors were visualized, and the animals were randomized on day 9. And you are seeing the ventral and dorsal view of few mice prior to the treatment, showing the establishment of tumor in the mice as fluorescence in the picture. So the more intensity of fluorescence means the more tumor burden in these animals. Next day, the mice were treated with either vehicle or saline, unmodified T cells, unmodified NKT cells, or CAR T cells, or ALA-101. Next slide, please. Then the tumor growth in the mice were monitored at regular intervals by looking at the bioluminescence image. An image on the top panel shows the tumor burden in the mice assigned to respective treatments, and the bottom panel shows the tumor burden after 3 days of the treatment with immune cells. As you can see on the image, a significant regression of the tumor was observed in the group treated with ALA-101, as indicated by the absence of fluorescence compared to other groups, displaying the anti-tumor activity of ALA-101 just in 3 days of infusion. And that is an amazing result that we saw in this experiment. So next slide, please. Then these mice were monitored for up to 90 days to see whether ALA-101 provides better survival rate in these treated animals. As you see in the figure, indeed, ALA-101 significantly increased the survival of these mice compared to that of CAR T cells and other controls. And this indicates that the in vitro response that we observed before can be recapitulated in a tumor setting in vivo. And taken together, we presume that ALA-101 has the potential to be an effective off-the-shelf therapy for CD19 expressing cancers in the clinic. Next slide, please. So during this experiment, we also noticed that 4 mice in the ALA-101 treated group showed relapse of the disease in the brain. And as you can see from the picture, in all 4 mice, relapse of the tumor, which is, you can see that it has the fluorescence, can be detected from day 24 to day 45 in all the 4 mice. And we were surprised to see that the relapsed tumor in the brain were eliminated over time around 52 days, without any additional dosing of ALA-101. And we refer it as a spontaneous secondary remission. And this data, in fact, infer that the ALA-101 could persist in vivo and continue to protect against the cancer in vivo. And with this data, we believe that the potential of ALA-101 could be leveraged for treating primary central nervous system lymphoma, where currently no standard of treatment currently exists. Next slide, please. So Arovella's iNKT cell platform has several advantages. Just to summarize on Arovella's iNKT cell platform, it has several advantages compared to other cell therapy modalities currently in use for allogenic off-the-shelf approach. The first and foremost thing is that we use iNKT cells from healthy donors to make the product, unlike some of the other sponsors who work with the iNKT cells, where they generate the iNKT cells by reprogramming stem cells. Since we use mature iNKT cells from the circulating blood, the polyclonality and the polyfunctionality of the iNKT cells are maintained. And there is less challenge in getting the product approved for clinical trial from a regulatory agency compared to that of a stem cell reprogrammed iNKT cell. And the second point is that we can also generate the cells with a higher transduction efficiency up to 60%, meaning that a high percentage of the isolated cells can be modified to express the CAR. We also start with a low number of iNKT cells as starting material, and so we only use less quantity of reagents to modify these cells. And as I have shown before, the efficient expansion from a single batch can yield multiple doses. The advantage is that this could potentially lower the cost of the product per patient. So from beginning to end of the manufacturing process, our product maintains the cytotoxic population of iNKT cells. In addition, preliminary data from the clinic shows that when dosed with unmodified iNKT cells up to 1 billion in the patients, these cells did not elicit graft versus host disease in patients with acute respiratory distress syndrome. And growing clinical evidence reveals that CAR-INKT cells are beneficial in the clinic in dose escalation trial of a similar product from another sponsor. With that, I would like to hand it over to Michael. Thank you.
Right, thanks very much, Minnie. I think that was an excellent overview of some of the science that's been generated to date, and hope everybody got a great indication of why we're so excited by the platform. And I'd just like to touch on a few more points quickly before we finish this presentation, but that's really important for us. When we were looking at technologies to develop, being differentiated, but having strong IP around the platform was really important. And so that's why we're delighted when we came in contact with Professor Tassos Karamitri at Imperial College London. And now we have that exclusive worldwide license to those patents for that manufacturing process. And it covers all disease areas, so all cancer types, and even things like infectious disease or autoimmune conditions potentially as well. So we've got patents for that filed in Europe, that's already granted, and also additional filings in the US, Canada, Hong Kong, and Australia, with patent life out until 2038. And of course, we also expect that as we progress and things do eventually reach the approval stage, that there's opportunities for additional exclusivity to be taken advantage of as well. Now, in terms of where we are at the moment, I think this is obviously a very important slide. So we are making great progress in terms of manufacturing. This is a key area for the development of the product and getting it into clinic. So we've already optimized the viral vector to engineer the chimeric antigen receptor. And we've used, I use the term here, regulatory friendly elements. So things that won't shock regulators, which are now well established across the industry. So that's done. Where we have identified high frequency donors, as many alluded to before, they are often low frequency. So we're collecting donors that will have the right signatures for us to manufacture the product. And that's something we'll continue to do so that we've got a whole suite of donors. Now, in terms of the manufacturing process itself, we're continuing to optimize that, and we'll do that through the first half of this year. And that's important so that we've got the process defined so that we can produce material that's ready for clinical trials. And in the second half of 2023, we'll finalize the scale up and generate the data required for regulatory submissions before producing material for clinical trials, and then ultimately getting this into patients. Now, just switch gears a little bit just to refresh people's memory on another exciting development that the company is working on, which is the partnership with Imugene to combine our platforms. So Minnie, I think, has done an excellent job of showing how potent and why we're so excited by the iNKT cell platform to kill CD19 producing cancers, and it will be developed as an off-the-shelf product. And that's going to be combined with Imugene's oncolytic virus, CF33. Excellent preclinical work to develop that virus to target specifically tumor cells, and then a further iteration of that product so that it actually causes the tumor cells to express or produce CD19 to push onto their surface. Of course, Imugene's already started its phase 1 trials for both the virus itself and also the virus that forces production of anti-PD-L1. So they'll already be collecting excellent safety data and biodistribution data, which we'll leverage for the co-administration. Now, in terms of from a mechanism of action point of view, so we have the oncolytic virus CF33-CD19 that gets injected into the patient. It will track and home to the tumor where it infects those cells and begins to replicate. Some of those cells will naturally be lysed because it is an oncolytic virus, but as it continues to expand and grow, it forces the tumor cells to produce CD19, and that will be then pushed out onto the surface of the tumor cells, which essentially become like little flags for the iNKT cells to recognize. So then we would expect to have a second administration of the iNKT cells. They're then able to find the CD19 on the surface of the immune cells, the tumor cells, and then start triggering their destruction. Now, Minnie mentioned also a few things before that not only do iNKT cells find and kill tumor cells, they influence the tumor microenvironment by destroying pro-tumorigenic cells like tumor-associated macrophages and myeloid-derived suppressor cells, but they also recruit or can cause a recruitment of endogenous host immune cells like T cells and NK cells. So we do see that there's a multifaceted approach here for killing solid tumors, which is exciting. It was nice that we already generated the in vitro data for that program, and we've based on that data, we've advanced and starting to work on animal models for a particular indication. So in terms of our shots on goal, we've got a number of potential areas where we can look to develop products. So certainly for CD19 CAR or ALA-101, we're developing that for non-Hodgkin's lymphoma or CD19 producing lymphomas. In combination with Imugene's onCARlytics platform, it does expand and open up the opportunity to target a range of solid tumors. And our DKK1 targeting technology, which we haven't had time to discuss today, but that also, the data supports using that as a treatment for potentially multiple myeloma, but also a range of solid tumors. Now, I'd just like to make the point here, so we can change the chimeric antigen receptors or missiles, and there's a number of different CARs that we could consider either developing on our own or in licensing, and they can be used across a range of blood cancers or solid tumors. So there's lots of scope to take the platform further. Now, in terms of our milestones, we do expect to have a very full 2023. And for ALA-101, the key next step for us is to work through the manufacturing process. So we're doing that over the next six to 18 months and intend to have that manufacturing defined, completed, and to have commenced phase 1 clinical trials for patients that have CD19 producing lymphomas like non-Hodgkin's lymphoma. Now, for Imugene and ALA-1 strategic collaboration, we'll be looking to continue to generate more proof of concept data to support not just the indication that we've selected, but potentially other solid tumor types as well, and then formally kick off IND enabling studies. And for DKK1 or the ALA-104 product, continuing to validate that target and then combining in parallel with our iNKT cell platform to generate proof of concept data. So as I mentioned in one of my earlier points, we do have a phenomenal team, which we've assembled, I think, over the last, say, almost 2 years, but 18 months or so. And it's continued to get stronger, which is great. So I think what pleases me is there's a lot of experience right through the way of research and development at an early stage, so deep scientific understanding through to translation of products into clinic and into commercializable products. And of course, at the transactional end, there's a lot of experience here in deal making and acquisitions and so forth. So it's a very great team to be a part of. A lot of experience in drug development and an enormous amount of experience in cell therapies, which is really important for the products that we're developing. And as I mentioned, we're continuing to strengthen the board. I'm delighted that Dr. Tom Duthie joined only a couple of weeks ago now as our chairman. He's already having a big impact at the company, which is great. Now, I think we can all appreciate that over the last 24 months, it's been a challenging sector, but I think I'd like to point out here that hasn't stopped completely any transactions in the cell therapy space, which is, again, exciting to see. And hopefully as the market stabilizes and picks up, we'll continue to see more transactions. But just this is a snapshot of some of the cell therapy transactions that we've witnessed more recently over the last 6 months to 3 years or so. And you can see that quite a number of them are actually at preclinical or phase 1 stages, and still with reasonable upfronts in the order of 70 to 200 million US. So there is still a fair bit of activity, which is good to see. Certainly for Arovella being, a preclinical and looking to get into phase 1, it does certainly open up the possibility of us having important discussions with some potential partners in the future. And I'd just like to finish with our financial overview. So we're currently valued at about just over 36 million. And we've got, as of December, our pro forma cash in the bank was 5.2 million. So we've got an excellent shareholder register. We're delighted to have Merchant as our largest shareholder, and also a number of other high net worths, but also small institutions that have been quite supportive, which is great. And so we're always looking to welcome new investors to the register, and certainly those that understand that there are risks associated with an investment into a biotechnology company. But of course, the rewards that can be achieved in an investment in this sector shouldn't be overlooked. And with that, I'd just like to say again, thank you very much to Minnie for presenting the science today. I think it was great to have that perspective. And it's certainly a big thank you to everybody that's taken the time out of their schedules to listen in today. Delighted to have you on the call. And Matt, I'll hand it back to you so that we can go through some questions and answers if there are any.
Great, thanks, Michael, and also to Minnie. Michael, look at you. You've stopped sharing your screen. Thank you. So we'll move on to the Q&A session, as you've mentioned. As I said earlier, the questions are based on those sent through via email and using the Q&A function within Zoom. So if anyone has a question off the back of all that, please feel free to use the Q&A panel to type it in and submit it on your screen. The first question we've got ready to go, I think it was touched on briefly, but just to reiterate, the secondary remission data in the mouse model is compelling. Can you comment on what this might mean for the product?
Sure. I might start with that, Minnie. Maybe you can chime in, but look, it was a pretty interesting finding from that study, right? And if you saw it in one mouse, you'd think, okay, maybe we'll put that down to chance. But we did see it in 4, so it's quite interesting. I found it hard to find other examples of that type of thing happening. But certainly from our perspective, I think it's, we'd like to delve into it a little bit further scientifically. But what it suggests early is that the iNKT cells were able to remain in the system in order to remove those tumors a second time. And of course, it does tell us that they're able to access the brains across the blood-brain barrier, and even potentially to eradicate a more solid tumor-like tumor in the form of something that's localized to the brain. So I think for us as a company, and it's something we started thinking about a little while back, but would like to explore further, is can we tackle diseases like primary central nervous system lymphoma, secondary central nervous system lymphoma, where the outcomes for these patients are really poor. There isn't much in the range in the realm of treatment for them, and the time frame at which things worsen and the patients ultimately succumb, it's months. So we do think that look, if you wanted to use an autologous T cell, it would be very difficult because you'd need to manufacture that product for the patient just in time, which would probably take too long. Versus having something ready to pull out of the freezer. We think there's big advantages, and not to go on too long, but I think there's a couple of other areas that I think would be really interesting. Certainly looking at, if Imugene's platform was also effective at targeting brain metastases, is that something we could also target using the combined therapies? Because I think that would be a really, again, for these patients, there's really not much support at that point. So it'd be a phenomenal product if it was successful.
I think you answered it well, Michael.
Thanks, Minnie.
The next question I have is, why are there not more companies working on iNKT cells?
Yeah, it's a good question. And look, it's something we do get asked a fair bit, to be honest, because you look at that slide I presented earlier and it's stark, right? There are hundreds and if not thousands of T and NK cell companies and associated clinical trials. And we still see, I think it's a handful of iNKT cell players and a handful of clinical trials, right? So it's still stayed small. One of the hypotheses we have is that The T cell complement is upwards of about 50% of our immune cells, so it's quite high. So they're able to get access to them. NK cells, I think it's in the range of 5 to 15%, so also quite high. Now, the frequency of iNKT cells for a good donor is quite low at 1%. So we think that potentially people have been intimidated by the manufacturing or the manufacturability. And again, that's been core to the licensing, was having the IP surrounding a manufacturing process so that we're able to produce a product that we can take to clinic and beyond. So I think that's a big factor. I think, probably scientifically, right, it's also they're less well studied, right? They're a far less well characterized and studied cell type. So we think that perhaps people have stayed away from it out of those reasons as well.
Yeah. And two reasons, as Michael said, it is one of the less frequent cell in the immune, in the system, and so it's very, so very so a few researchers are trying to, expand these cells, and one of those is there will be licenses technology from, and he's well known in that NKT cell space as well. So that's primarily the reason why many people are not, trying even, like scared to do this, since we have the capability to, and we have actually established, and we know it is capable of scaling up. We are confident that we can go ahead with this product.
And I think just to add there, right, it's an important point that the IP that we've licensed really does address a key challenge around iNKT cells, right? That provides an advantage for Arovella in being able to develop this cell type, even though it is difficult to work with.
Great, thanks for those answers. The next question I have is, although it's always hard to give definitive timeframes, ideally, when would Arovella hope to be commencing an ALA-101 phase 1 trial?
Excellent question. So I think, look, the key factor for us is manufacturing, right? And we're making great advances on that already, but that's the step that we want to get right. And so we're going to make sure that we're doing it in a way that gets us into phase 1, but it means that we're not in a position that after we've completed phase 1 that we're having issues expanding to the US. So we're doing that work at the moment throughout the course of this year. So we've already got our hands around the vector, which is great. That's one of the key components for the therapy. And the next steps are to optimize and scale up the actual manufacturing of the cells. So we're making good progress. And I think before the end of the year, we'll have a better handle on what that looks like. And hopefully we've got a good manufacturing process established.
And look, this question might be very much along the same lines, but what is the potential time frame for collaborating with IMU in dosing human patients?
Yeah, that's a good question as well. And I think, look, the reality is for that, we still need to generate proof of concept data. I think what we've got early is encouraging, and we'll continue to build upon that data, both in terms of breadth, but also going into animal models. And in presuming that goes well, which we expect that it will, we've also then got to go through the formal pre-IND enabling studies. Now, just to be clear, so Imugene have already started phase 1 with their oncolytic virus, so we'll be able to leverage that data of distribution and so forth. We will need to start phase 1 with ALA-101 and do some dose escalation work before we can start a phase 1 with Imugene. So we will need to start that and probably have a couple of cohorts dosed, and we'll work with the regulators to see if we can't initiate the combination study with Imugene at in a more expedited pathway, provided we've got good safety data initially. But there's a few things to do before that one gets into clinic.
Right. The next question is, in manufacturing from single cell to multiple or billions of cells, what medium are the cells grown in?
Minnie, do you want to take that one?
Yeah, sure. So just to clarify that we are not growing from a single cell. We are actually collecting a few, we can, depending on the scale of manufacturing, either it's research or, for clinical grade manufacturing, we are collecting a bunch of cells from the donor blood, which is not single cell. So we have millions of cells to begin with, and these cells are expanded in a media conducive of the proprietary media that is conducive for the growth of these human iNKT cells, and that is, is cultured for several days, as I shown in the data set. Hope this clarifies that question.
Yes, thank you. The next question is, in layman's terms, if everything goes well, are we potentially looking at a cure for cancer with this iNKT cell therapy, and across how many different types of cancer that exist in the world today?
Yeah, look, I think I'm not going to say it's a cure for cancer just yet, right? But that's obviously what we're aiming to do. We're not aiming to have marginal improvements here. This is a real aiming to be a shot at goal and having a big impact. And I think when you look at the cell therapy data that's been generated to date for Yescarta and Kymriah and patients 10 years on for hematological malignancies, they are cancer-free. There's a lot of potential in this space. So the data that we've got to date suggests that iNKT cells will also play a really an important role in this space. Now, one thing that Minnie touched on a moment ago is this concept of CD1D expression in various cancers, right? And so when we have our CAR and CD1D targeting particular malignancies, we do see better activity than what we would see for T cells, and that covers a range of different blood cancers, CLL, marginal zone lymphoma, mantle cell. So we do see that there's a huge potential for iNKT cells in that space. Now, we also know there's a range of solid tumors, probably not as many as blood cancers, that also produce CD1D. And so they're certainly cancers that will be a focus or an area of interest for us. And one of those in particular is glioblastoma, which is one of the nastiest forms of brain cancer. So look, I think there's a lot of shots on goal. We clearly need to get a lot more data as we get into clinic. But one of the other things I'll just quickly touch on is the option that we entered into with University of North Carolina last year. That also gives us another shot on goal to even enhance the platform one step further. And if that's the case, if it's going to be an attractive product for solid tumors, we think this would just go another step further in making it stronger. So again, very excited to see where the technology goes and how it performs in clinic. And that's certainly going to be the key focus for us over 2023.
Great. And a reminder, if there is any further questions, please feel free to type them in, because this is coming to maybe the last one. Can you comment on the use of the PTX platform toolkit to expand the cancers that can be targeted with your technology?
Yeah, look, it's a good question. And we've spoken with Steve, and look, I think there is scope to do it. It's just, there's a limited number of resources and opportunities that we need to explore, but certainly it's something that, I think particularly the one that influences the way the effector population of the cells could be of interest. So look, I think anything like that where there's scope to cross-pollinate and work together, we'll certainly explore. And ultimately we'll let the science dictate whether or not those things progress.
Great. Well, that does conclude the session for today. So thanks again to you, Michael, and to Minnie and Nicole for your contributions. That was really informative. If anyone has any further questions, obviously feel free to contact us using the details on the ASX announcements, various channels available to you. But that concludes the webinar and Q&A for today. And thanks to everyone for taking the time to join.
Thank you.
Thank you.
So.